Hi everyone, I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and subscribe the channel. In the last session, we ended it with steps of glycolysis. In today's session, we will continue with the same. In this session, we will see fructose 1,6-bisphosphate undergoing aldol condensation. Then we will see glyceraldehyde 3-phosphate showing dehydrogenation. After that, we will see how fluoride irreversibly inhibits glycolysis, and hence fluoride is added to the blood while taking blood for sugar estimation. Then we will see how aerobic condition varies with anaerobic condition in pyruvate metabolism. Then lastly, we will see energy yield from glycolysis. Good morning everyone. Let's start the session. Step 4 of glycolysis. The 6-carbon fructose 1,6-bisphosphate is cleaved into 3 carbon units, glyceraldehyde 3-phosphate and another molecule of dihydroxyacetone phosphate, that is DHAP. Since the backward reaction is an aldol condensation, the enzyme is called aldolase. This reaction is reversible. Step 4a of glycolysis. Dihydroxyacetone phosphate is isomerized to glyceraldehyde 3-phosphate by the enzyme phosphotriose isomerase. Thus, the net result is that glucose is now cleaved into 2 molecules of glyceraldehyde 3-phosphate. The step 4 and 4a are together called splitting phase. In this diagram, we can see triose phosphate isomerase in which dihydroxyacetone phosphate is reversibly converted to glyceraldehyde 3-phosphate. Glycerol portion of the neutral fat can enter into glycolytic and gluconeogenic pathways at this point. Similarly, neutral fat synthesis glycerol is required, which can be derived from glucose through DHAP. Step 5 of glycolysis. Glyceraldehyde 3-phosphate is dehydrogenated and simultaneously phosphorylated to 1,3-biphosphoglycerate, that is 1,3-BPG, with the help of NAD+. The enzyme is glyceraldehyde 3-phosphate dehydrogenase. The product contains high energy bond. In this diagram, we can see glyceraldehyde 3-phosphate dehydrogenase in which glyceraldehyde 3-phosphate is reversibly converted to 1,3-bisphosphoglycerate and conversion of NAD+ to NADH. The oxidation of the aldehyde is an exergonic reaction that derives the synthesis of high energy compound 1,3-bisphosphoglycerate with high phosphoryl group transfer potential. The enzyme is cistenyl CA group at the active center and is inhibited by iodoacetate. During this reaction, NAD+ is reduced to NADH. Step 6 of glycolysis. The energy of 1,3-BPG is trapped to synthesize one ATP molecule with the help of bisphosphoglycerate kinase. This is an example of substrate level phosphorylation, where energy is trapped directly from the substrate without the help of the completed electron transport genes. In this diagram, we can see bisphosphoglycerate kinase in which 1,3-bisphosphoglycerate is reversibly converted to 3-phosphoglycerate and conversion of ADP to ATP. When energy is trapped by oxidation of reducing equivalents such as NADH, it is called oxidative phosphorylation. Step 7 of glycolysis. Glycolysis. 3-phosphoglycerate is isomerized to 2-phosphoglycerate by shifting the phosphate group from third to second carbon atom. The enzyme is glucomutase. This is a readily reversible reaction. Step 8 of glycolysis. 2-phosphoglycerate is converted to phosphoenol pyruvate by the enzyme enolase. One water molecule is removed. In this diagram, we can see steps 7 and 8 of glycolysis. 3-phosphoglycerate is reversibly converted to 2-phosphoglycerate with the presence of enzyme phosphoglyceromutase. Then this 2-phosphoglycerate is converted to phosphoenol pyruvate with the enzyme ethynolase and magnesium and removal of one water molecule. The high energy phosphate bond is produced. The reaction the reaction is reversible. Enolase requires magnesium, and by removing magnesium ions, fluoride will irreversibly inhibit this enzyme. Thus, fluoride will stop the whole glycolysis. So, when taking blood for sugar estimation, fluoride is added to blood. And if not, then glucose is metabolized by the blood cells so that lower blood glucose values are obtained. Step 9 of glycolysis. Phosphoenol pyruvate, that is PEP, is dephosphorylated to pyruvate by the enzyme pyruvate kinase. First, PEP is made into transient intermediary of enol pyruvate, which is spontaneously isomerized into ketopyruvate, the stable form of pyruvate. 1 mole of ATP is generated during this reaction. This is again an example of substrate level phosphorylation. The pyruvate kinase is a key glycolytic enzyme. This step is irreversible. In this diagram, we can see that phosphoenol pyruvate is converted into pyruvate with the presence of enzyme pyruvate kinase and conversion of ADP to ATP. The pyruvate kinase is a key glycolytic enzyme. This step is irreversible. The reversal, however, can be brought about in the body with the help of two enzymes, pyruvate kinase and phosphoenol pyruvate carboxykinase, and the hydrolysis of two ATP molecules. Step 10 of glycolysis. In anaerobic condition, pyruvate is converted to lactate by lactate dehydrogenase or LDH enzyme. LDH has 5 isoenzymes. The cardiac isoenzyme of LDH will be increased in myocardial infarcts. On the other hand, in aerobic conditions, the pyruvate is sent to citric acid cycle for complete oxidation. The end product is anaerobic glycolysis is lactate, which enters the Cori cycle. Coming to significance of lactate production, step 5 and 10 are coupled. In the fifth step, for each molecule of glucose entering in the pathway, 2 molecules of NAD plus are reduced to NADH. The availability of coenzymes inside a cell is limited. Therefore, the step becomes a bottleneck in the whole reaction sequence. For smooth operation of the pathway, the NADH is to be reconverted to NAD+. This can be done by oxidative phosphorylation. However, during exercise, there is lack of oxygen, so this reconversion is not possible. Therefore, the cell has to couple with some other reaction in which NAD+ is regenerated in the cytoplasm itself. Hence, pyruvate is reduced to lactate. The NAD+ thus generated is utilized for uninterrupted operation of the pathway. In RBCs, there is no mitochondria. Hence, RBCs derive energy only through glycolysis. Hence, where the end product is lactic acid. Here, in this diagram, we can see that step 5 and step 10 are coupled. Glyceraldehyde 3-phosphate is converted to 1,3-bis-phosphoglycerate with the presence of enzyme glyceraldehyde 3-phosphodehydrogenase and conversion of NAD+ to NADH. On the other hand, pyruvate is converted to lactate with the presence of enzyme LDH and once again reconversion of NADH to NAD+. Coming to energy yield from glycolysis. During anaerobic, that is the efficient oxygen deficient condition, when one molecule of glucose is converted to two molecules of lactate, there is a net yield of ATP, 2 molecules of ATP. 4 molecules of ATP are synthesized by the 2 substrate level phosphorylation, but 2 molecules of ATP are used in the step 1 and 3, hence the net yield is only 2 ATP. In this table, we can see energy yield from glycolysis in case of anaerobic glycolysis. Step 1 contains hexokinase as an enzyme and it disrupts 1 ATP. Step 3 contains phosphofructokinase which disrupts 1 ATP. On the other hand, 1,3-bis phosphoglycerate kinase generates 2 ATPs, and step 9 pyruvate kinase generates 2 ATPs. So in total, 4 minus 2 is equal to 2 ATPs are generated in case of anaerobic glycolysis. The whole reaction is summarized as glucose plus 2Pi plus 2 ATP gives 2 lactate plus 2 ATP. Hence, but when oxygen is in plenty, the 2 NADH molecules generated in the 3-phosphate dehydrogenase reaction can enter the mitochondrial electron transport chain for complete oxidation. As each NADH produces 2.5 ATPs, this reaction generates 5 ATPs. Thus, when oxygen is available, the net gain of energy from the glycolysis pathway is 7 ATPs. Hence, the ATP from the glycolysis is different in anaerobic and aerobic conditions. In this table, we can see energy yield from glycolysis in case of aerobic glycolysis. Step 1 contains hexokinase as an enzyme and it disrupts 1 ATP. Step 3 contains phosphofructokinase as an enzyme and it disrupts 1 ATP. On the other hand, glyceraldehyde 3-phosphate dehydrogenase generates 5 ATP. Step 6 1,3-bis phosphoglycerate kinase generates 2 ATPs, and step 9 contains pyruvate kinase which generates 2 ATPs. Thus, total 9 minus 2 is equal to 7 ATPs are produced in case of aerobic glycolysis. The pyruvate is later oxidatively carboxylated to acetyl-CoA, which enters into citric acid cycle. Complete oxidation of glucose through glycolysis plus citric acid cycle will yield a net 32 ATPs. Here ends our session. Kindly like, share and subscribe the channel. Thank you.